Matt, are you ready to get started? And Ryan, you're doing all right, ready to get started? And Ted, you're still sounding all right and you can hear me okay? All right. For everybody online, thank you so much for joining us today at Hatfield's research seminar. My name is Cinnamon Moffett and I'm the research program manager at Oregon State University's Hatfield Marine Science Center located in Newport, Oregon. And I am your host for today. If you haven't joined us before, your mics and screen shares and videos are turned off and disabled, but we're hoping that you will put any questions you might have into the chat whenever you have them, and we'll get to them at the end of whoever is presenting at that time. But please feel free to put those questions into the chat so that we can interact with our presenters today. I wanted to also let you know that we are recording today's seminar and it will be posted up on our website, which I just put into the chat, in a couple of days. So if you missed something, you want to see it again, or you want to share it with somebody else, feel free to go to that website that I just put into the chat, and the video will be there soon. Couple of quick announcements for next week's seminar, which is September already, September 2nd, is Megan Considine from CEOS's Marine Resources Management Program at Oregon State University, who's been working with ODF&W Steve Rumrell at Hatfield. She will be giving a seminar about shell boring polychaetes in the Pacific Northwest oyster aquaculture industry. So that will be a really interesting one. So please join us next week for our seminar. If you want to learn more about any upcoming seminars or any of Hatfield's events, please go to HMSC's homepage, scroll to the bottom, and there's a calendar of events there where you can find all the login information that you might need. The other thing I wanted to promote real quick is that we are looking for an OSU student to help with seminars moving into hybrid format in the fall. So if you are interested in helping me out for a few hours a week, this is a paid position and the position closes on the 31st of this month. So you've got until next Tuesday. I'll put the link to the job posting into the chat here in a few minutes and you can check out the job posting. But interested in having a little bit of help. So if you are looking for an opportunity and you like coming to seminar anyway, you'll be able to make a little bit of money by helping me out. So check it out. But for today, we have a double header. So we're very excited and very fortunate. And both of our speakers have been invited today by Ted DeWitt, the former branch chief of EPA here at Hatfield. So I'm actually going to hand this whole show over to Ted, who's going to introduce our first speaker of the day. Ted?
Hi, thanks, Cinnamon, and hi, everybody. It is my pleasure today to introduce my colleagues, Matt Harwell and Ryan Gillimet, to present back-to-back short seminars. I'll introduce Matt, and then he'll speak for about 20 minutes-ish, and then after some questions, we'll transition to Ryan's seminar. Matt, Ryan, and I all work for EPA at the Office of Research and Development at the lab here on the Newport campus. Matt is the new branch chief of the lab here. He's the boss. He's a senior ecologist with experience in estuarine ecology, seagrasses, wetlands, and restoration. His other expertise and where his research is focused these days is on adaptive management and governance, stakeholder engagement, connecting science to decision-making, ecosystems for decision support, and strategic science communication. His talk today will be on translational science and strategic communication in environmental research and management. Go, Matt.
Great. Thanks, Ted, and thanks, Cinnamon, for inviting me. Hello, everybody. My name is Matt Harwell. It's very nice to join you this afternoon. Before I switch over to my slides, which are going to be focused on more of a social science lens, I'm going to put in the chat box the link to the EPA's portfolio for the Marine Science Day this past spring, if you want to learn more about the active work that we're doing in the EPA lab just down the hall. So I did that, and now I'm going to share my slides. Let's see, hang on a second. And that did not work. I practiced this, I promise. Hold, please. Okay, fantastic. So thanks. I appreciate your coming today. So this isn't going to be a traditional marine science talk. As an ecologist, I've been working in social ecological systems for several decades now. Many of us spend time thinking about the applications of our work and how our work can, quote unquote, inform management decisions. But what I'd like to do is sort of push the envelope a little bit on some human dimensions work. And today I wanted to introduce some core elements of translational science and strategic science communication that I've had the opportunity to work on recently in my time at ORD. And again, here the focus is on translation and on strategery. So hopefully this will give you some creative ideas that might be relevant to your work. I am going to sort of do the disclaimer at the bottom of the slide that I am indeed talking at EPA. Can somebody confirm that they can see my screen? I don't see the border showing that that's visible.
It looks great, Matt.
Great, thank you so much. I appreciate that. Okay, so a translational science perspective helps us think about how to turn data and observations into information. In particular, for supporting decision-making in the health arena, it's called for making interventions. Overall, so that we can work towards improving the environment, improving human health and well-being. A couple of components of a translational science definition are sort of shown on the left there with the quotes from Austin. So translational science focuses on producing products that are translated for users and audiences. It also focuses on involving researchers, implementers, and stakeholders. And so a concept that's relevant here is that of co-development of research vision and implementation that helps focus on addressing well-defined and often unmet needs of partners and stakeholders. You can also think about translational science as focusing on a research along a spectrum, and I'm going to introduce this framework in a minute here. Starting with fundamental questions and working your way through sort of how that's developed. So what I want to do is introduce a number of types of frameworks from non-ecology world, a lot of social science world. The first one I'm going to introduce is actually from the environmental public health. And so that's this one here from Pettibone. This is a translational research framework from the environmental health sciences world. So again, might not have run across this if I had not been sort of working with folks outside of the ecology disciplines. The idea here is to think about your work from multiple components on a spectrum, starting with fundamental questions, working your way through applications, implementation, full-on practice, and then an intentional process for understanding the impact of that work. And a description of each of these are sort of here. And when we talk as sort of classical natural scientists, we typically play in these first two spaces, this fundamental research kinds of space, and also in this space of doing structured and predictable testing and applications. The next level down, though, talks about focusing on testing in real world settings and looking at adjustments based on the differences that are happening in the real world. The next tier then focuses on practicing and sort of moving from an implementation of tests into real world practice. And then an area that most people don't spend a lot of time talking about is sort of thinking about the impacts at this larger end of the spectrum. These are the types of products that you typically think about in terms of what you're generating as a scientist or as a researcher. And so using this kind of a framework helps you think about, how do I take my fundamental questions research and maybe move it a little bit further down the road and think about applying it? Or maybe I want to, think about how I'm applying my work, but really what I want to do is get it to this next level where it's actually being implemented on the ground, real world and established practice. So this kind of a translational framework helps you think about how you can move across scales, as well as build dimensionality of research within a given scale. Maybe I can add some dimensionality to my existing fundamental research. And so I'm going to give you an example of how we might think about this. So the example I'm thinking of is a project that looked at how to use ecosystem services assessments. So this is assessing the benefits that nature provides, ecosystem services, as part of an environmental decision-making context. And this particular project looked at examining tools that were involved in those assessments and looked at applying them in a Superfund environmental cleanup context. And so this original research design was sort of set in this sort of third tier space, right? Looking at testing tools and 2 case studies. But it also touched upon other elements of this framework because it was designed that way. So it was built upon previous ecosystem research, ecosystem services related research. And it was also built using existing tools that had already been sort of developed in this application conversation space. By thinking about it from this perspective, as that project was underway, they also talked about how they were testing the efficacy of their work. We often think about it in terms of identifying gaps or areas of next steps in our research design. And in this particular case, they realized that they needed to actually move back across the layers into further development that was particularly needed. Another element of this project that was particularly interesting from a translational science perspective is that it was co-developed with the end clients, the customers, the stakeholders in mind. And so in this case, as they were working through this, one of their end clients were Superfund site managers, so those responsible for the environmental cleanups themselves. And in the course of this work, they discovered that essentially there was a need that had not been previously identified. And that was actually providing more foundational information about the benefits of nature that could be useful for Superfund site managers. And that alone translated into development of a new type of product, science product, that hadn't been previously identified. And so in this particular example, a single project actually figured out ways to touch upon multiple elements of a translational science framework. In this particular case, the project was a little small, so it didn't quite get into this impact conversation space. And it's not too uncommon to sort of see that as a challenge in terms of most projects don't really get to that point of evaluating the impact. But it is something that we should be thinking about how to do more of. All right, so another element that I want to introduce is this systems thinking kind of perspective. So I'm a classical ecologist. I know what systems thinking is. From a social science construct, it's a little bit different. It's not thinking about all the different components of an ecosystem and how they're all connected together. It's actually thinking about social systems. It's thinking about public health systems. It's thinking about ecological systems and how to work towards integrating those elements together for a larger, bigger cause. There are a couple of different kinds of examples of how you might think about applying a systems approach to your research or your science or your question at hand. One example is thinking about how to potentially integrate diverse data streams across different disciplines, across different spatial and temporal scales, right? We're now entering this world of big data. We're entering this world where you can do some pretty fancy statistics much easier than when I was a graduate student. And so a systems approach is a way to think about how to integrate different kinds of diverse data streams. Another example, and the one I'm going to go to next, is talking about how we might think about expanding the way we think about conceptual models of a system. For example, adding dimensionalities to a model, or turning an ecological conceptual model into a social ecological. And so what I want to introduce is this concept of system maps. And so we typically think about using a system maps approach like this diagram on the left as a way to define what's going on with a particular problem or a particular place. So this is a very classic example. This is a conceptual ecological model of an estuarine system. In this case, it's down the road from me in Florida Bay. Very typical kind of model, drivers and pressures at the top, what's going on in the ecosystem in the middle, and the types of endpoints, environmental or ecological endpoints or valued ecosystem components at the bottom. Very classic kind of structure. You see this a lot. You see other derivations of this with different kinds of frameworks, a Dipser framework, et cetera, et cetera. This super coarse structure itself might be transferable from one place to another. But much beyond that, the components themselves may not be. So where this conceptual model for Florida Bay might be relevant to Biscayne Bay just down the road, it might not be exactly relevant to, your backyard here, right? And so we have a challenge that the way we think about system maps are a little translational, but maybe not as translational as they could be. So the question is can we learn something from the social sciences that might help us think about that? And as a quick side note, we do see this kind of challenge in mathematical model transferability as well. And I'm not going to talk about it today, but ORD has done some recent work about tackling the issue of assessing ecological model transferability. And if you're interested in learning more about that, please let me know. I can point you in the right direction. Ted DeWitt would be happy to sort of help you learn more about that. Okay, so back to this idea of how can we think about system maps from a slightly different kind of perspective that might push the envelope on translational science. And so what I want to do is turn to the social sciences again. And in this case, I want to introduce the concept of ethnographic methods. So these are a suite of qualitative and quantitative social science methods that are used to observe and learn interactions in and around a particular setting or a particular environment. And so the idea here is to develop a systems map that's not issue specific, but it's designed to be translational and transferable. So this one I'm going to introduce here from Katie Williams is what's called a neighborhood model. The elements in this model are generalized and built upon research from the public health arenas and the geography arenas. And the idea is to think about organizing the model on how the different elements contribute to a common endpoint for an environmental decision-making effort. And so for an example, what I'm going to show you is how to think about environmental decision-making from different perspectives, perspectives from different actors. So for example, the city is interested, so that's an actor, is interested in trying to improve the economic opportunities and infrastructure in its development plans. Whereas the neighborhood that might be involved is interested in something slightly different. Perhaps they want improvements to neighborhood trails that are in and around the area. Very typical kind of thing that you would see in a community, including in a community like Newport. So a translational science framework then helps us make distinctions between these different kinds of perspectives. So the first two major components in a neighborhood model is what is common to us as sort of ecology types. So we understand the notion of having things from the built environment in our models. And we understand this notion of having structural dimensions in the way we think about our models. But it adds a couple of new elements that we probably really hadn't thought about before. So one is this human environment relational dimensions conversation space. And to be honest, we're starting to get a little bit more into this kind of work, right? This is kind of what we think about when we throw around the term human dimensions kind of work. And a fourth space is this area called personal experiences. And so I'll show you an example for that city and the neighborhood and the wanting to build trails versus trying to get a better feel for how we can have more economic development for the community. Again, this is a systems map that's designed to be transferable. So when you populate it out, it looks a little something like this. So the elements from the built environment in this particular case, you can sort of see what those building blocks are. There's specific building blocks of structural dimensions, natural features. So that rings a bell with me, right? In the human dimensions area, so there's sustainability, resilience. We're spending a lot of time talking about these kinds of concepts right now. But in this personal experiences space, a classic sort of ecology or environmental decision-making space doesn't really think about these kinds of things quite as much or quite as often. And so the question is can we use these ideas to help us advance the way we think about conceptual models? Okay, so given that, what we want to know is can we take this kind of way of thinking about a system map and actually make it transferable? And the answer is essentially yes. And what I'd like to do is show you how we might do that in an ecological model setting. And there we go. Slight delay on my. Okay, so can we use the way we think about conceptual ecological models and this concept from the social sciences, this framework approach? Can we try to stitch these two worlds together? And I think the answer is yes. And so I want to sort of walk you through that. So this might be familiar to a number of you. This is a 2007 conceptual model for your backyard, actually, right? So this is the factors influencing nutrients and response variables. So there's the report citation there that Cheryl and crew developed back in 2007. And I'll show you the legend sort of in a few minutes. Now, this was built for a specific context, right? Nutrients and response variables associated with nutrient criteria. But could we potentially think about how we might think about using and leveraging or adding dimensionality to this to turn it into something more of a social ecological systems kind of concept? So I'm not passing judgment on this awesome work that was done for nutrient criteria perspectives, but thinking about this dimensionality. All right, so let's see how that might play itself out. So this is the legend for those graphics, and there's about 8 or 10 of those kinds of icons, and I'll toggle back. Yep, there's 8 or 10 icons right there. Okay, so those 4 components from the neighborhood model. So built environment elements. Yep, I see 2 of those here, pulp mill and estuarine shoreline development. So we do indeed have some examples from the environment, the built environment. We do see some of those already in this existing model. Human environment relational dimensions. Yep, it looks like there's a couple of those here as well. There was a component identified related to logging and a component related to recreational uses, including recreational fishing uses and recreational boating uses. So that's good. So there's some continuity there in those ideas. About structural dimensions, that was a 3rd component in this in this neighborhood model concept. There are a couple pieces that fall into the structural dimensions space, but they turned out to be exactly the same thing as the built environment elements. So there might be an example where we might use this social science framework and think about, are there other aspects of either social, are there other aspects of the built environment that we might want to capture, or other aspects of the structural dimensionality that we might want to capture in advancing this kind of a conceptual model and something that's more of a social ecological model. Let's look at the last one. So the last one was personal experiences. So this is the fuzzy part, right? For those of us that are classic natural scientists. And then it turns out, not surprisingly, in a nutrient criteria kind of conceptual model, we don't actually see that here. But the notion, though, that we use this social science framework to think about how we develop our conceptual models, this might create an example where we would go down a specific exercise to look at a gap analysis. Are there areas of personal experiences that we think we might want to capture in terms of adding dimensionality? So that's the kind of thing that we might think about for an area of future development and future exploration. Okay, so those are 2 pieces I've sort of introduced so far. I've introduced sort of that continuum diagram, and I introduced this notion of a social science framework that we might think about the way we think about modeling. Here's another way we can sort of think about translational science. And what we're doing in EPA is we're using a lot of our efforts thinking about our science from the perspective of how do we inform different parts of the decision-making process. And in fact, we're bringing in, again, from the social sciences, the concept of structured decision-making as a formal entity itself. So excuse me. So this is a way of thinking about the decision-making process in a very systematic manner, starting at the very beginning of a decision process, talking about the context, working your way through understanding the objectives of what you're trying to accomplish, talking through how you might think about the alternatives that might be involved in your decision. How might you identify and estimate the consequences of making a decision A versus a decision B, which then leads itself to an evaluation of those trade-offs, which then feeds the decision process that allows you to implement. Theoretically, you get the opportunity to monitor and review. And if you are able to connect the dots back together, you just created that adaptive management kind of concept where we get into this sort of what I describe as the shampoo bottle effect, lather, rinse, and repeat, where you get the opportunity to go around and around. So what we're interested in is trying to take our work and framing it in the context of this structured decision-making cycle, because that itself is a translational tool, right? So maybe my work is relevant to a NEPA decision process. I could map my work directly onto the NEPA steps, but then I'm done, right? So then that work probably can't be translated into a non-NEPA kind of context. But if I map it onto this process, this SDM process, I already know how NEPA maps onto SDM cycle. I already know how the Superfund cleanup process maps onto the SDM cycle. I already know how these other processes. And so we can use this SDM cycle, the structured decision-making cycle. We can think about it sort of as a Rosetta Stone, if you will, or a decoder ring. And so that's another area of translational science that might be relevant for how we think about making our work relevant for decisions on the ground. And if you're interested in learning more about structured decision-making, I put a couple of references in here that are really good. There's a classic book on SDM as a collective whole, and then a couple of really key foundational materials on the concept of structured decision-making in environmental contexts. Okay, so why don't we do this? The next piece I want to do, okay, so what I'd like to do then is talk about how we might further develop out components of structured decision-making from a translational science perspective, specifically with the idea of advancing environmental decision. And so the next piece I want to introduce to you, so this is, I guess, the fourth piece on our journey, is this notion of advancing the way we think about stakeholders and how to engage stakeholders and how to prioritize stakeholders in an environmental decision-making context. So building from the fields of business and other disciplines, so again, bringing in things that are not in the natural sciences, some folks in ORD has developed a suite of criteria for how do we think about prioritizing stakeholders that are involved in some kind of environmental decision-making context. Surprisingly enough, or maybe it isn't surprising, there hadn't been a systematic way to consider stakeholders from a natural sciences kind of perspective. In fact, in my former life, focused on ecosystem restoration, quite often the environmental components were based on how loud somebody pounded their fist on the table at a particular meeting. And so the idea here is how do we think about getting out of that model and getting science to have a seat at the table? So we work to develop these types of criteria to help us sort of navigate the stakeholder universe. And so I'm just going to spend just a minute on them briefly. So the first two criteria focus on the magnitude and probability of impact a decision might have on a particular stakeholder. Another pair of criteria that are involved in prioritizing stakeholders has to do with the level of influence and interest that a stakeholder has in a particular decision context. This one looks at the immediacy issues. So whether it's something is either urgent or temporal or a spatial immediacy issue. So that's these here, whether you're immediately adjacent in time or in space to the issue at hand. There's a criteria on economic interests. So again, this is way outside of the way I think as an ecologist. Here's a couple of that focus on things like rights and fairness in terms of elements for evaluating stakeholders. Rights often have a legal connotation with them. Again, something that natural scientists don't often think about. And then the last criteria that we developed was this notion of focusing on underrepresented and underserved populations and spending a little bit of time exploring those as well. So the notion is, by having the systematic criteria, we can then talk about how to get the stakeholders at the table at the right level and the right ability to engage in an environmental decision-making process. And so let me give you just a quick hypothetical example. So what you could do with these criteria then is you can think about how to weight them for a particular stakeholder group or weight them across stakeholder groups so that you can think about what to bring it to the table. So in this particular example, flood mitigation plans and decision-making needs, and potentially including environmental restoration components. What might be involved in those kinds of decisions? Well, we certainly see flood mitigation conversations happening in the Pacific Northwest coastal communities, right? So how do we think about that? Well, if you're thinking about it from a planning perspective, how might you identify what those elements are for that flood mitigation process? Perhaps these are the kinds of criteria that are the most important ones at hand. And so maybe these would be the 5 that you would focus in on the most. And that then might lead you to better understand how to engage the right stakeholder groups to help you with that planning perspective.
Matt, I'm just going to give you a quick time check. It's 4 o'clock.
Great, thank you so much. And then there might be a different way you might think about it from a communication kind of perspective. And so that might be something that would be relevant to think about. The last piece I wanted to do is, I'm going to skip 2 more real quick. The last thing I wanted to do is introduce this kind of conversation space about turning data into information. And sometimes there's a challenge in working with lay audiences to understand the difference between data and information. And how do you think about making things helpful and trusted? So here's a social science sort of construct methodology for thinking about whether information is helpful or whether information can be trusted. And what you can do is think about the way you've got information being fed and how you think about that data being shared. So maybe in this community, focusing on a hypothetical virus problem, maybe your information is more helpful and more trusted from a university perspective than, say, a state scientist perspective. Or maybe you think about the information coming from the federal officials might be slightly different than the way you think about it from a local or a state officials. Or maybe you want to think about where that information is actually reaching the community and whether it's actually coming from friends and family as opposed to other sources. And so by looking at a construct, something like this, you might have the ability to think about how could you take what you want to do. I'm a Fed scientist. Maybe I want to move myself here. How might I take and think about moving my work from a dimensionality or utility kind of perspective? So those are the kinds of things that I wanted to sort of introduce with you all today in terms of how to think about a social science kind of perspective. I am going to stop there and hit the done button. So I appreciate your willingness to go on a journey with me today.
Thank you so very much, Matt. We appreciate it. I think there is a lot that we could dig into in that short presentation, but I want to make sure we give Ryan a little bit of time. So for folks, if you have questions for Matt, go ahead and put them in the chat, and if we have time, we'll get to them at the very end. But if we don't have time today, Matt's going to be spending some time with us. So
absolutely,
we know how to find him. So I'm going to hand this back to Ted. Ted, can you introduce our next speaker? And thank you, Matt.
Yes, indeed. Thanks, Matt. Our next speaker is Dr. Ryan Guillemette, who's a postdoctoral researcher with expertise in aquatic microbiology. Ryan is working at the EPA lab here and has been with us for about 1.5 years. His focus in aquatic microbiology is in pathogen ecology and microbial aspects of water quality. Ryan received his PhD from Scripps Institute of Oceanography and went on to a one-year postdoc at MIT before he came out to join us. His talk today will be on the microbial ecology of rainwater. Over to you, Ryan.
Hi, Ted. Thank you for the introduction, and thanks for giving me some time today to introduce myself to the Hatfield community. Today's talk will be on rainwater microbiology and with some an outline of some work that I'm doing here, some of the preliminary results. But I did want to first share some background that I have in marine science. As Ted mentioned, I was at Scripps for my PhD. And while I was there, I was in Dr. Farouk Azam's lab and was looking at coastal ocean microbial ecology, biogeochemistry, and pathogen ecology. We had a couple of papers that came out in 2020 on Vibrio, including with a collaboration with OSU. So former OSU postdoc Blake Ushijimi and Claudia Haas, who's in the Carlson College of Veterinary Science. And just putting that up there. So if any folks, are interested in having a virtual coffee and talking shop, I'm definitely interested in integrating with the community here. But I'm going to switch gears a bit and talk about some freshwater stuff, and just want to introduce the topic of rainwater microbiology in general. So it's a relatively understudied field of science in terms of microbiology. When you think about marine science and freshwater science, specifically like lakes and rivers and streams, the literature has a substantial amount of research that's been published, probably thousands of papers in that field, whereas rainwater microbiology has a handful, about 4 or 5 that have been published to date. And it's interesting because microbes are so ubiquitous on Earth, and they often play a major role in biogeochemical cycling of critical elements, specifically carbon. And of course, as we all know, they can serve as pathogens. So I was just curious, what's the fundamental baseline of microbes in falling rain? This is rain falling directly from the sky before it's contacted any surfaces. And some questions that came to mind as I observed all the rain falling here and the wheels got turning was, what is the significance of these rain-associated bacteria in general? How about the basic questions like their abundance? Are they viable, metabolically active? What is their taxonomy? Where are they coming from? And there's been some work that's shown that there are multiple sources where airborne, atmospheric, and rain precipitated microbes can be aerosolized from crashing waves, like for example, like we have here on the active coast in Newport, agricultural fields, as well as various sources of dust and surfaces in our terrestrial environment. And once these rain deposited bacteria hit their new habitat, are they viable? Are they able to survive? And you start thinking, what happens when you transport microbes from the atmosphere into rivers, lakes, the ocean, agricultural fields, and are there any implications for pathogens to disperse? And I'm specifically in this case interested in human pathogens. There is some literature showing that the rain can transport plant pathogens, various Pseudomonas bacteria that can infect tomatoes, for example, and some other crops. But of course, there's also some implications for rainwater harvesting. So the EPA has an active program in Cincinnati looking at the microbial water quality from rain harvested water sources. And their emphasis really is on the microbial ecology and water quality in the collected water. So the water that's already hit surfaces and gone into some type of reservoir for holding. And I'm backing up a step and asking, well, what's going into that in the first place? And the interest there stems from this, periods of drought that the Western US has been experiencing. And locally here in Newport, as many, if not all of you know, we've had recent restrictions on water use for various reasons, but the lack of rain isn't helping there. So people have proposed, for a long time that rain harvested water can be a supplement to community water demand. And I was just wondering if you take the average, rainfall in Newport, which is approximately 71 inches, and you plug that into a simple model, how much water would you get from the average home? And so a typical standard home on the low end, a conservative estimate would be a 1,500 foot square roof, put in that 71 inches of rain. It's about 66,000 gallons of water per year coming from an individual home. And what does that mean? Well, I called up the city and asked them, what is my usual water consumption rate? And they have me at about 4,000 gallon a month, which is about 48,000 gallon a year. There's 2 people, my wife and I, in the home. So if we were, for example, to collect all of the rain that fell over the course of a year, we would have met our entire water demand with 20,000 gallons of water left over, which is substantial. So I think there's, water use, water rights, municipal water regulations is a very political and touchy topic. So I won't comment on that, but I think the calculations show that there's potential for rain to provide us a sustainable source of water in our local communities here, at least. So it begs the question for me, what are the implications of microbial water quality here, and specifically what can I do to help address the effort? And that's to look at the pre-contact microbial ecology, the parameters that people might consider when they want to assess the quality of water that's collected. And I've set out a series of collections on the roof here at the EPA building to get the pre-contact water. So this is water that hasn't touched any surface, hasn't touched the roof, hopefully hasn't been in contact with any trees or anything on its way down. And asking simple questions. What's the abundance? Using direct counts, DAPI stained bacteria under the microscope. Are the cells metabolically active? Using this quick chemistry technique, which I'll briefly describe, it's essentially spiking the rainwater sample with an amino acid analog that gets incorporated into actively growing or protein synthesizing bacteria cells. And that is then stained and can be directly observed under the microscope, as shown by these cells here in green. I don't know if you can see my cursor or not, but these small green cells are actively growing bacteria from collected rainwater here in Newport. And also wanted to know what the community was, so sequencing the DNA, and also the active community. So again, using tracers, taking a nucleotide, basically an analog for thymidine, that gets taken up by the cells and integrated into the bacteria's DNA. And that newly synthesized DNA in actively growing bacteria will contain the bromodioxyuridine, BRDU, in it. And you can extract that using a complicated immunocapture technique that I won't go into, and you can sequence the 16S genes to determine your active bacteria taxa. And actually this method was partially developed and pushed in the late 90s and early 2000s from one of the labs here at OSU, the Giovannoni lab, as well as Farouk Azam's lab at Scripps. So anyway, lastly, I wanted to know what type of materials in this rain that could potentially support or affect microbial growth. So that includes total organic carbon, the TOC, nutrients, as well as pH. And I have time just to show a few preliminary results. The study is still underway at the moment, but I'm finding very low bacteria abundance, which is not unexpected. The few studies that have been published show values of approximately 10 to the 2 to 10 to the 5 cells per mil, with some people thinking the 10 to 5 is suspiciously high. My direct observations are showing 5 times 10 to the 3 bacteria cells per mil. And that's like 2 to 2.5 orders of magnitude lower than you might expect on the coastal waters here or the estuary waters. So it's really low. And surprisingly to me was that I had only found about 1 doubling. So the cell population doubled or increased twofold within a 24-hour period, which suggests, at least for this particular sample set, that there wasn't favorable growth conditions for the bacteria. So in line with that, I want to state that the pH was low, 5.0. That's within the acceptable range, the normal range of rainwater, but it's quite low for supporting microbial growth. And the total organic matter, the total organic carbon rather, was 20 to 30 micromolar carbon, which is extremely low compared to marine water, typical fresh water, and estuary water. So I think what's going on is that you have some active population of cells that are in the samples, but with that low pH and low organic matter, there isn't much room or favorable conditions for them to grow to substantial numbers. So in terms of implications for rainwater harvesting, I've been working with some folks at the EPA in Cincinnati to better understand and define some of the microbial water quality parameters. So what we should be looking for, testing, and what type of information from these baseline studies could be useful to those efforts. And I want to run a series of experiments to test directly whether known human pathogens, such as E. coli, Vibrio cholera, for example, and a suite of other isolates, are capable of growing in collected rainwater. And with that rainwater, it would test directly, but then also manipulate the pH, the organic carbon, and the temperature to see if any of those variables are either suppressing some of the growth, or if you were to alter any of those variables in the environment or in collection reservoirs, whether you might expect growth in that end. Another experiment that might be interesting to do in collaboration with the Cincinnati folks would be to collect rain and study, so the pre-contact water freshly falling from the sky after it's been deposited onto a surface, for example, like a roof, and then stored like in a cistern, and then once it comes out for end use, and just to follow the microbial ecology step by step. So that's the basic outline of a study that I've been working on, some of the preliminary results and the next steps. And in the interest of time, I'm going to cut it there so we can have some questions, and also some questions from Matt's talk. But I'd like to first acknowledge my advisors and supervisors at the EPA here in Newport, Cheryl, Ted, and Matt. Also Jay Reichman, who's at the EPA in Corvallis, Jay Garland at the EPA in Cincinnati, T. Chris for technical assistance here at the lab, and as well as the EPA for a small award that's funded some of this research, the Pathfinders Innovation Project. And from there, we'll go back to Cinnamon and or Ted to help us navigate any questions that might be out there. Thank you very much.
Perfect. Thank you so much. So for folks online, if you have any questions for either of our presenters, go ahead and put them in chat. And Matt, the first question is going to come to you. It says, nice presentation. Have you thought about how well this fits into the fisheries management? It seems like the council process in many ways is a practical example of what you presented on. Do you have any experience with the council, and can you speak to this?
Yeah, thanks, Greg. That's a good question. Yeah, so these kinds of concepts I've been talking about are indeed relevant to fishery council approaches, although I'm not so sure about the specific one that you're referring to. Most that I am familiar with are typically regional in nature. They involve participatory governance by a suite of actors and stakeholders, and they often tackle a range of environmental decision-making things. And so there's a range of types of effectiveness across those types of governance structures, whether it's fishery councils or otherwise. Sometimes those are often driven by, charismatic leaders making that process successful or effective. Other times it's because they've put together the right governance model to make those things effective. And typically most of those successful governance models really does play with this dabbling of both the social and natural sciences in trying to set up the way to tackle these environmental decision-making questions. So I hope that was a good kind of answer for you, Greg. It was a little nebulous, but hopefully that was helpful for you.
Thank you very much. And Greg, if you want to follow up, just go ahead and type it in the chat and we'll get there. This is going to be a little fun because our topics are so different, but I have a question coming into Ryan. So Ryan, you had suggested that you were interested in understanding the source of these bacteria. Now, I know that that's not something that you've had a chance, I don't think, to look at, but can you talk a little bit more about that?
Yeah, absolutely. So I think first off, there's a body of literature that might be of interest to, I didn't catch who the question was from, but to whoever asked the question on aerosolization of microbes. So there's a group of atmospheric chemists at Scripps Institution of Oceanography and UCSD in the Prather Lab. And they've pumped out a number of papers showing bacteria, algae, potentially even virus, active enzymes, for example, that have been released coming from sea spray aerosols. So my guess, my assumption would be that here in Newport, we have such high, rates of crashing waves that you probably have a lot of either microbes aerosolized from there. There's also research that stems from places around the world really on aerosolization of soil-borne microbes. So these are microbes that are attached to little dust particles or loam or sand, and the wind picks them up and carries them, and then they might get encapsulated by the rain, either while they're suspended freely in the air, maybe 10, 20, 100 feet above, or even further up in the atmosphere, but that's not known. So those are some areas, those are the most likely sources.
Yeah, that kind of speaks to the question I was going to ask you, was just about variability and whether we're able to notice what is, like you were saying, is it after windstorms, after, those kind of events where we might see a spike in what we're seeing in the rainwater?
Right. Yeah, that's a great question and comment, really. I think it's the type of experiment or study one could run. So testing when different environmental conditions there. So it's difficult to control because you have all these major processes happening around the region with the wind and the rain moving. So it's hard to pinpoint down. But one thing we've talked about is using microbial source tracking. So looking to see if you have specific DNA markers in your samples that could be an indication of the source that they came from.
Okay, questions are coming in now. But before I do that, Matt, Greg just wanted to say, yes, thank you for answering my question. That was a good response. Ryan, this one comes back to you. What are your expectations for potential growth of pathogens in stored rainwater? Is there any specific threats that have come up in your findings?
So expectation's tough. Conditions can be variable. I think things to consider would be when you have stored rainwater, then you have potential for algae growth, you have potential for contamination of DOC. And if you're supplying the microbes, even if they're at low initial abundance with food, with organic matter to grow on, then you could have a potential explosion of growth there. And in terms of pathogens, it's likely that pathogens would be coming from like excrement released by birds on roofs, for example, or any sort of like downstream from sewage processing plants, these types of things. So just keep in mind that one bacterium can grow exponentially with the right organic matter. However, with that said, it's very unlikely from my experience that you would have human pathogens coming from the wild, right? So from a breaking wave ending up in a cistern and growing, because pathogens usually follow hosts. And if you don't have hosts where those microbes are being generated from, they're probably not pathogenic to humans.
And so Ryan, this next one is for you as well, and it just expands a little bit on what you touched on there. But Cheryl says, I was surprised at how high some of the nutrient values and was wondering whether the airborne transport of bacteria might support the nutrients also being transported from ocean into the rain.
Yeah, Cheryl, that's a good point. So for those of you who don't know, Cheryl is actually my mentor. This data is brand new, so we haven't had a chance to discuss it. So I'll leave part of that conversation for later. But yeah, I was surprised there's a lot of nitrogen in there, and even some silicic acid actually, which was a surprise, which suggests maybe coming from the ocean. But yeah, there's enough nitrogen there to support bacterial growth. And it makes you wonder if microbes are active in the atmosphere or in the rain, if they're actually cycling those elements en route to their destination.
Well, we can thank Cheryl for putting you on the spot in public.
Yeah, no worries.
She apologizes. I don't know if you see the chat. Thanks, Cheryl.
I'll pull it up. It's all good. I appreciate it.
I think there's a lot of interest in your work. I think that's what you're getting from all these questions. If anybody else has questions, go ahead and throw them into the chat. And if it's a little quiet, we'll give everybody a second. I just wanted to kind of promote the idea that Matt is probably, and I'm just going to hint at this, going to be working with us when we do our research summit as well. So if you are curious about following up with some of the ideas that maybe he presented, that might be a good time to learn a little bit more from Matt. All right, I've got a request from Michael to ask a question. So hang on just a second, Michael, I have to find you. All right, Michael, go ahead.
Ryan, I just wanted to meet you voice a little bit because I was very intrigued by your research. I grew up in Africa where a lot of people live off water harvested from the roots. And I guess I was fascinated to learn from you today that there are so few studies about that because much of the rest of the world lives like this. And indeed, a lot of us in Oregon that in a rural situation do that because our summers are so dry. And it's a really interesting place to live in terms of water. So I guess I was just intrigued to hear that there's just so little information, and yet, you know, many people in the world live like this. Do you have any comment to that?
So Ryan, I know that Michael was hard to hear, but our question was around, since so many places around the world do depend on collecting rainwater for many purposes, can you talk a little bit about why there hasn't been a lot of study around the bacterial load in those systems around the world?
Yeah, sure. I appreciate the comment and the question. There's a couple of things there. So first, there have been studies on the water that has been collected. For example, you get a reservoir filled with water, people take a sample and study that. There are fewer studies, far fewer studies on understanding what is actually falling into those reservoirs, the pre-contact rain. And I'll comment on why I think there are few studies on that. It's very difficult to study low abundance microbial ecology with the techniques that are available to us. So that's the major reason. You need a certain microbial mass to sequence the DNA. You need a certain amount of microbes to observe them under the microscope. So when they're at 10 to 2, 10 to 3, 10 to 4 cells per mil, it's much harder to detect them. And with recent technology that's become available, that's become easier. So I think you might find more of that. The other component of it is it's tough. Like, it's hard to collect water that hasn't been in contact with anything. When you go out, if you stand in the rain and take a look, water's flying all over the place, the wind is whipping, and it becomes very difficult to control that sample collection. And it's frankly can be unpleasant to be outside in the rain when the wind's blowing and it's cold. So I don't think as many people are chomping at the bit to get out there and take those samples. That would be my guess. But again, thank you for the comment and the question, and we could talk further on that if you have other questions.
Thank you. So it looks like we are running close to the end of our time. So I just wanted to really encourage everybody who's online, if you are interested in following up with either Ryan or Matt, please do so. I know that Ryan has been here at a time when we haven't been able to interact face to face. So please reach out to him if you are curious, if you have ideas, if you want to share. Ryan, we have a whole team of geneticists that are here on site, and I'm not even sure that you've met them yet. And so, yeah, please help make those contacts and connect with everybody. I'm being asked, Ryan, if you can see the chat to put in your email, that would be really great, into the chat box.
Happy to do so.
And yes, Matt, we will bring more speakers to us, and it's great that we're able to collaborate. So Ryan just put his email into the chat, so please feel free to reach out to him. For everybody online, thank you so much for being here and being a part of this crazy double header of 2 concepts in a very short amount of time. So thanks for everybody. Hope to see you next week. Really great talks. You're getting applause in the chat, virtual thanks. So for everybody online, thank you so much for our 2 presenters. This is the beginning of conversations and not the end, but thank you for being here with us. And we'll see you another time. Thanks, everybody.
Thanks,
Bye.
everybody.